Next presentation is by Ruth Sedlak and co-workers, entitled Detection of HHV-6 Bioactivation in HCT Recipients with Inherited Chromosomally Integrated HHV-6A by Doppler Digital PCR.
Hello, good afternoon. My name is Ruth Sedlak. I am from the University of Washington, the Molecular Clinical Virology Laboratory. I work with Keith Jerome and our collaborators at the Fred Hutchinson Cancer Research Institute. And so today I'd like to tell you about a new assay we've developed for the detection of both HHV-6A and B integrations by droplet digital PCR. So as we all know, after hearing talks today, inherited chromosomally integrated HHV-6 is a condition that affects about 70 million people worldwide. HHV-6 integrates into the chromosomal regions of different chromosomes in this condition. And this condition can really confound testing for active HHV-6 infection. This is an example of a case report showing engraftment of stem cells, and as the stem cells engrafted, the HHV-6 viral loads increase. So by quantitative PCR, you cannot tell if this is an active infection or whether it's associated with ICI HHV-6 unless you have prior knowledge of the donor's integration status. And so for this reason, better diagnostics are certainly required to study not only the clinical significance of integration, but also to improve HH6 monitoring in transplant patients. And so today we had a nice overview of quantitative PCR this morning and how that works. So I want to contrast that with how droplet digital PCR works. So in quantitative PCR, you use a primer and probe set to get amplification of your target of interest, and you use a standard curve to get a quantity. But in droplet digital PCR, you get an absolute quantification based on endpoint PCR. So it's the same TAC-MAN primers and probes that you would use in a quantitative PCR assay, but you cycle the endpoint, so you can actually count positive droplets and negative droplets, and in that way, you're getting an absolute quantity. So we have previously we developed a clinical assay to identify ICI HHV-6 based on an assay targeting HHV-6 and RPP30, which is a cellular reference gene. And so this is just an example of what a droplet plot would look like, where you have droplets that are positive for HHV-6 only, droplets positive for RPP30 only, and then double positive droplets compared to completely negative droplets. And this is an example of what a plot would look like on a person with integration. So this is a cell line example where there's one HHV-6 for every cellular genome. And that is the ratio you would expect in a patient whose blood cells you test in integration. You'll have one HHV-6 genome for every cellular genome. And so we've taken this test a step farther by developing it to speciate between HHV-6A and B in a single test. So our previous clinical assay targeted U67 with one primer set and one probe that targeted both HHV-6A and B indiscriminately. But now we've changed this to targeting U94, where we have one primer set for HHV-6 and two probes, one that's specific for HHV-6A and another specific for HHV-6B, where there's 5 nucleotide differences in the probe. And so a droplet plot for this would look something like this. If you had an ICI HHV-6A cellular sample, your plot would look like this, where you have, it would look like that, where you have the droplet populations forming more of a square. But we've been able to, by varying the fluorophores on the probes for HHV-6B, you can shift the droplets. And so in one test, you can determine if you have integration and whether it's from an A or a B virus. And I mentioned on the previous slide that we tested this assay and our previous clinical droplet digital PCR assay, and the clinical results are concordant in terms of determining the ratio of HHV-6 A or B to cellular genomic equivalents. And so to utilize this test outside of just the clinic, but also in research, we wanted to determine what proportion of HCT patients who are CI HHV6 positive or receive cells that are ICI HHV6 positive will reactivate an HHV6B infection, because now we actually have an assay where we can determine that by just looking at the DNA. And so the rationale behind this is that, of course, we know that around 30 to 50% of transplant patients reactivate HHV6B, and that reactivation of HH6A is rare. And we know that active infection in the background of integrated HHV6 would be overlooked with traditional qPCR testing, just because you cannot see the active infection in the background of all of the genomic DNA. And so we applied our droplet digital PCR assay to detect HHV6B reactivation in patients with integrated A. And so this is an example of an experiment I did to show that we could get sensitive detection of HHV6B viral particles in the background of an ICI HHV6A cell line. And so I spiked serum samples with a cell line known to have integrated, HHV6A, and then also added HHV6B viral particles to that serum in a 10-fold dilution series. And so this is showing that we could detect a constant background of the integrated A while detecting a dilution series of HHV6B virus with a sensitivity of around 95 copies per mil of serum. And so we used this assay on a cohort of patients that Josh Hill will tell you more about tomorrow, but they screened thousands of HCT donors and recipients for ICI HHV6. And out of around 7,000 screens so far, they found 72, 18 of which are positive for ICI HHV6A, and 54 are positive for B. So out of those 18 patients that had ICI HHV6A, we took 16 of them and found that they had weekly serum or plasma samples available for up to 8 weeks post-transplant, and tested those by our droplet digital PCR assay. And so I'm showing the results here. 19% of those, so 3 out of the 16 ICI HHV6A HCT recipients did have a reactivation event for HHV6B. And shown here in red is the HHV6B viral load, and in black is the HHV6A from the integrated form. And in 2 of these patients, patient 1 and patient 3, the donor cells were the source of integration. And so you can see that at the pre-transplant time point, there is no HHV6A detected because the recipient's cells don't have any A. They receive it from the donor, and so you can see that as engraftment occurs, the HHV6A viral load increases in both of these patients, whereas in the patient that received, that was integrated and received related stem cells from the donor, there's a constant background of ICI HHV6A. And in each of these patients, there was 1 reactivation in a single sample of HHV6B that we could clearly detect with this assay. And so I will mention that this ddPCR assay is available in our CLIA-CAP certified facility. So this is a clinically available test for ICI HHV6A and B that only requires blood cells as opposed to hair or hair follicles or nail trimmings. And we do have unique access to specimens from a large cohort of integrated patients, thanks to our collaboration with the researchers at the Fred Hutch. And this data really does point towards the need for more advanced diagnostics to identify HHV6B active infections in the background of either integrated A or B. So a limitation of this assay in particular is that we still cannot detect active infection of B in the background of integrated B, which, as you can see, is still a large proportion of the integrations. And so to get around this, we are currently working on mRNA assays, because that would clearly be the best method. But that does require a lot of upfront education in the clinics in terms of getting samples that are appropriate for RNA testing, because currently most samples that we get would just have degraded RNA, and so there would be no point in having an RNA assay. So right now, this is our best alternative. And with that, I'd like to acknowledge Keith Jerome, who is the division head for all of the virology division goings-ons. And Thuy Nguyen is the research tech who did a lot of this work, and we collaborate closely with Joshua Hill, Danielle Zaire, and Michael Book on all of these patient samples. And then I do need to recognize Biorad Laboratories as well. They have provided us with a more advanced reagent to use for this sort of testing that should be available soon commercially. So I'll take any questions.
Thank you.
So if I understood, it looked like 3 out of 16 individuals with HHV-6A reacted their B, and the reactivations were really just only a few times did they reactivate. So the question is whether having integrated HHV-6A reduced the rate of reactivation. In other words, we heard this morning it's usually about 50%, and it's usually very, multiple time points would reactivate. So can you speculate a little bit about that?
Right. Well, we're not really sure. We were kind of surprised to only see one positive sample out of all of the samples available, and we're not really sure why that might be. I'm not sure. That's my answer.
Hello. I have a question concerning the strains that you have studied to design primers and probe in U94, especially for HHV-6A.
What strains we use to create that assay?
Yeah.
So just the PubMed GenBank sequences that are available, and this U94 assay is actually one that was published in one of Danielle Zaire's papers from 2002, and it's one that we've been using in the clinic since then. So it could certainly be updated with
which strain exactly?
Is it U1102? Is that the A strain?
1102, yeah, 1102, yeah, because this strain is different from the majority of the strains circulating in the population. It's a very special strain, and we have sometimes problems to design consensual primers for this strain and the other. And then, did you try to compare with other strains, A strains?
So this assay,
no.
10 years ago when they validated
U94?
No, we have not sequenced any U94s from clinical isolates, but that is something we're interested in doing. I've talked to a couple of you about doing some whole genome sequencing for HHV-6. If that's something that interests anyone and you have clinical isolates available for us to sequence, we are interested in doing whole genome sequencing using Illumina next-gen instruments. So we all need more sequence data for HHV-6A and B.
And just to complete, with using a real-time PCR, there was developed a method to distinguish A from B, and with a difference up to 3 logs. And it's possible using real-time PCR.
Yes, and we have done that clinically as well.
But not when you have a difference of viral load more than 3 logs.
Exactly. And so in this background of high genomic HHV-6, it's difficult to discriminate an A and a B when you have the B reactivation with qPCR. That's what we found in our hands.
It depends on the quantity, relative quantity,
Yeah. But that's not an issue in the droplet digital PCR. Yeah, it discriminates at all log differences.
I just want to make a quick comment. Coming back to the previous question, there's actually a fair amount of reactivation which is very short-lived and only one single week detectable. So of those 40 to 50%, I think at least 10 or 20% are in that category. So I think it's a very small sample size, 3 out of 16. I wouldn't make too much out of it. I think, yeah.
A few years ago, this is a long time ago, I guess, we deposited in GenBank a number of 6A U94 sequences from German isolates that might have been used when designing those primers.
Right, because that's different than the whole genomes. Correct, yeah. So I have to give credit to Mei Li Huang in our lab. She was the one who designed the U94 assay, and that's what's in Danielle Zer's paper, and that is what we used for this, since we have lots of historical data in our laboratory using this assay.
Okay, thank you.